Hey guys, this is part one of genetics. So to start off with, we obviously would look at Gregor Mendel and the experiments that he did. And he used what he called a pea generation, which are true breeding purple and white pea plant flowers. Now in my drawing over here I don't have purple, or I wasn't thinking I may have it, but I didn't use it. I've got blue instead. But that represents the purple flower. And he crossed them with what we call true breeding white flowers. Those are, true breeders are organisms that only breed a particular trait. So he would allow these purple flowers, in this case blue, to reproduce several generations. They always produce the same color flower. Same with the white. So he took those and he crossed them, and he gets what he called the F1 generation, which are all purple flowers. Every one of them. Every generation. And he didn't just do this once or twice. He did this hundreds and thousands of times to get his results. So he said every time he did this, he always got the exact same result. Then he allowed these guys to self-fertilize or self-pollinate, and he looked at their offspring, and he called that the F2 generation. And he noticed that he once again had both the purple and the white flowers. And when he started counting these up, he ended up with a ratio of 3 to 1. 3 purple to 1 white. So he called the trait down here that was most prominent, he called that trait dominant. He said that trait dominated the other trait up here in the F1 generation. And the trait that disappeared but then reappeared down here in the F2s, he called recessive. A couple other terms that goes along with genetics that we need to know to help understand a little bit more about Gregor Mendel's experiment. The first is allele. An allele is the alternate form that gives a different trait. We're talking about the same gene here. For instance, the gene that codes for flower color in pea plants, Gregor Mendel would say that there were 2 alleles. There was a purple allele and there was a white allele. So the allele is just that alternate form for a particular trait. Then when we're learning how to write these and understand them, there's a couple of terms that we like to use. The first is homozygous. In a homozygous individual, that is 2 alleles. The 2 alleles are the same. So, for instance, sometimes we designate these with letters: big A, or a little a. These would have been the pea generation plants that Gregor Mendel was using. So, if he was using purple flowers and white flowers, he said purple was dominant to white. So he would have used big P. So we would say that the purple parent was big P. It was homozygous dominant. The white flower plant would have been little p. Notice it's still the same letter as the dominant one, but it's the lowercase. So lowercase p, which would have been homozygous recessive. Another term is heterozygous. Heterozygous is that the alleles are different. For instance, big A, little a. So in Gregor Mendel's experiment, that would have been the F1 generation. You would have had, they were all purple, they would have been big P, little p, or lowercase p. So I've got that down here for you. In Mendel's experiment, the P generations were big P, and little p. So that would have given you the purple and the white. These were homozygous for the traits. In the F1, he would have gotten all heterozygous individuals, big P, little p. And then in his F2, this is where it gets a little bit more complicated. He would have had both homozygous dominant, that should be big P, I don't know why I've got F's there, heterozygous, which are big letter, little letter, and then homozygous recessive as well. So when we look at traits in genetics, one of the things that we like to do is look at what is the probability of getting particular offspring. And to do that, in Bio 2, we primarily use 2 different things. We use either monohybrid crosses or dihybrid crosses, and that's about as big as we get. In a monohybrid cross, we're looking for a heterozygous for 1 gene of interest. So for instance, in our plants that Gregor Mendel was using, this would have been the cross of the F1s to make the F2 generation. So we get big P, little p, heterozygous, times a heterozygous. We put that in a table that we call a Punnett Square. And a Punnett Square gives you the possible offspring that might be produced. It doesn't guarantee that any of these will be produced. It says this is the possible offspring. So you can see here that we take one of the P's and put them here, the other one and put it here. What we're doing there is looking at the possible gametes that would have been formed. So we say if the individual is diploid, the gamete would be haploid, meaning it would only have one of the chromosomes. So if there's a big P here, then there's a chance of a gamete getting a big P. Little p here, then there's a chance of a gamete getting a little p. Same with this individual parent. We see that up here. So we put this in the Punnett Square. We take this gamete and we put it in these boxes. We take this gamete and we put it in these boxes. And so on and so forth. So we get 4 possible offspring. One of them being big P, 2 of them being big P, little p, and 1 of them being little p. Now what we know from dominant and recessive is that this one would have been purple, this one would have been purple, this one would have been purple, and this one would have been white. And that lines up with the ratio that Gregor Mendel got in his experiment, a 3-to-1 ratio. We can complicate that and make what we call a dihybrid cross when we're looking at heterozygous for 2 genes of interest. So the standard would be big A, little a, big B, little b, times a big A, little a, big B, little b. So when you think about the possible gametes, remember we're assuming here that these are not on the same chromosome. But you would say this big A could be in a gamete with this big B. This big A could be in a gamete with this little b. This little a could be in a gamete with the big B. And the little a could be in a gamete with the little b. So you've got 4 possible gametes. Both parents would give you 4 possible gametes. So your Punnett Square would end up being 16 possible offspring. And that's a really big table. I'm not going to draw one for you on the tables or on the charts here. We will be doing some examples of these in class though. So how do we know what we're talking about or what we're looking for when we're looking at genetics? One of the key terms that we use is called a genotype. In a genotype, that's looking at the genetic makeup of an organism. So, for instance, that would be that they are big A, or big A, little a, or little a, little a. That's the actual letters that they have on their chromosomes that we give them. Another way to look at that is the phenotype. In a phenotype, that's the physical traits exhibited or the picture. So in Mendel's experiment, in his P generation, remember we said that would be big P, or purple, little p, or white. The genotype is the big P, or the little p. The phenotype is the purple or the white. In genetics, there's also a couple laws that govern what we do. The first is called the Law of Segregation. In the Law of Segregation, each allele in a pair separate into different cells. And you can think of this as what happens in anaphase 2. In anaphase 2, remember, that's the separation of the chromatids. That's what we're talking about. Because each of those chromatids would have the allele present for a particular gene. So those are going to be pulled into different cells. That's the law of segregation. The law of independent assortment, which we have already talked about before, is that each pair of alleles separates independently.
Miss Umbarker, can you call Miss Tucker in the main office? Thank you.
Independently from each pair of alleles on the different chromosomes. So if an allele's on a different chromosome, a different set of homologous chromosomes, then we would say they segregate or assort independently of each other. And this we saw in metaphase 1 of meiosis. When these chromosomes line up, they don't have to line up in any particular order. They can line up in whatever order they want. So the genes on this homologous chromosome are not connected to the genes on these homologous chromosomes. And they're going to line up independently of each other. Now, when we're looking at that in a real life demonstration, to see whether it's dependent assortment or independent assortment, you can take this little example here. So if you have, let's say, a seed that is either round and blue or wrinkled and green, and round and blue are the dominant ones. So if these are the P generations, then your F1 generation is they're all round and blue and they're heterozygous. If those traits are on the same homologous chromosomes, meaning they're connected, then that means they're dependent on each other. And they're dependent, the only gamete possibilities would be big R big B and little r little b. So whenever these would self-pollinate, you would end up with a simple monohybrid cross. And that would give you a 3-to-1 ratio of 3 blue circle ones to 1 green wrinkled one. Most traits, however, that we're looking at don't have this ratio. Instead, they are over here. So in this case, you have a dihybrid cross. So you've got 4 possible gametes, and we'll do some examples of this in class. 4 possible gametes, which gives you 16 possible offspring. And what you find is 9 of those offspring will be round and blue. 3 of them will be wrinkled and blue. 3 of them will be green and round. And 1 of them will be round and wrinkled and green. So you get a 9 to 3 to 3 to 1 ratio. This is what we see most often when we're comparing 2 traits, because that's independent assortment. Most genes will assort from each other as if they're on different chromosomes. And we'll talk about that a little more in lectures to come.